Hey, thank you, Sarah. My name is Jonah Kuhl. It's really a pleasure to be here with you all and looking forward to the next talks. As Sarah mentioned, we'll take questions from Carlos's talk as well as the next three at the end as a part of the panel. So please continue to input those into Slido. And then
the next four, because I'm there too.
So actually, yeah, so there was one change, which is that Jean-Laurent Casanova will be speaking, but he'll be speaking as a part of the second session. So just so that everyone's aware to kind of stay tuned for his talk, but we'll skip over that and he'll be speaking in that next session. And as a result, we will also go to break just a little bit early and plan to start that second session at 12:30 Eastern time. So without further ado, I'll introduce Ben tenOever from the Icahn School of Medicine at Mount Sinai.
Thanks, Jonah. Appreciate it. Thanks for the invitation to be here. Actually, I feel like this is a nice talk to follow Carlos's great clinical work. It is a slight change in subject matter, but what my lab is working on is essentially establishing animal models for SARS-CoV-2 infections in order to test a variety of vaccines and therapeutics for a number of different organizations. And so I'm going to just run through some of that data today in a relatively short period of time. So I think this probably is lost on everybody. I mean, not lost on everybody, but clearly this is a very serious problem that continues to develop. I wanted to just discuss, the take home, the high level view of what is supposed to happen when we are infected with a virus. And so, the quick answer to this is, as many of you probably know, is viruses make aberrant products that the cell doesn't normally make. This would include RNA hybrids, so double-stranded RNA, kind of depicted here in this cartoon view, or an RNA where the 5' end isn't modified the way it would be if it were a cellular message. So an exposed moiety like a triphosphate should not be there. But these are telltale signs of virus infection. And so we've evolved to recognize these structures through a variety of pattern recognition receptors. These include pattern recognition receptors that live on the outside of cells as well as those that live on the inside of cells. And essentially what happens is upon recognition of those structures, it sets in motion the activation of transcription factors, which are things that are going to turn on the genes that are important for fighting off virus. And those really come in these two flavors. So the activation of these two different families of transcription factors called NF-kappa B or interferon regulatory factors, these really induce the so-called call to arms. That is, they tell all the surrounding cells at that site of infection that infection is imminent, that it has been already detected in at least one cell. And it would be important for all the neighboring cells to fortify their defenses in an effort to slow infection down. And that's mediated by this interferon signaling, which I don't really have time to get to in a 10-minute talk. The other thing that those same cells are doing is they induce the activation of a broader family of transcription factors called NF-kappa B, where NF-kappa B alone drives the production of a lot of secreted proteins, some of which have chemoattractive properties. We call these chemokines. So what they're doing is they're creating a very high concentration of this secreted protein or secreted proteins at the site of infection that dilute out the farther they go. And what this provides is essentially a trail to bring in neutrophils and monocytes and other resident adaptive immune cells to the site of infection. And they're really the heavy hitters that are going to be responsible for controlling it. So the idea is that these two work together, that the call to arms slows the infection down at the local site while you call in for the reinforcements to truly neutralize the infection altogether. Now, every virus that you've ever heard of, if you've heard of it because it causes disease, it does so by blocking many of these pathways. And one of the unique things about SARS is not its capacity to block the call to arms. In fact, that's quite common for influenza, for rabies, for respiratory syncytial virus, for polio. All of these viruses had strategies to block the interferon system because otherwise they wouldn't cause disease. And so it's not surprising that SARS-CoV-2 has already been documented to block much of the interferon response. But what is unique about this virus is that that blockade seems to be limited to this family of transcription factors called the IRFs, and it leaves NF-kappa B essentially unchecked. And so as a result, virus infection stops the call to arms, but leaves the call for reinforcements on. And this is essentially the recipe for what ends up being COVID, because you're not controlling virus replication at the site of infection, but you are bringing in all these adaptive responses, which are going to come to the scene of infection only to find virus everywhere. And so long story short, we initially did some work in ferrets that we decided was a poor model because ferret replication is really contained in the nasal turbinates, whereas the Syrian golden hamster, it actually goes into the lower respiratory tract. And so I'm going to go through this pretty quickly to make sure I stay on time. But needless to say, if you come in with 100 infectious units or 10 to the 5, so a lot, and you infect a hamster, you see a pretty nice trajectory of disease. So obviously we prefer the lower inoculate. So just at 100 PFU, you can appreciate that you have, an established infection on day 1 that begins to climb and really peaks at day like 6 and 8 when 100 PFU are used. And then the hamsters start to recover, virus titers go down, and we see the production of neutralizing antibodies. So I'd like to think of hamsters as your average 20 to 30-year-old, and they're modeling what it's like to get virus. They definitely get infected. They definitely feel the infection, but this is by no means lethal, and it doesn't induce respiratory distress and wouldn't cause hospitalization should they actually be a person. Alternatively, and this is really, actually just something for those people who are doing this type of work in labs, I would say that in general, the community is using too much virus in their animal models and their cell models. So very commonly we come in with 10 to the 5, and I hope you can appreciate that you lose this dynamic of kind of virus goes up, virus goes down, and there's going to be a response to that trend. Whereas at 10 to the 5, you really have this like plateau, and then the virus is gone, and you lose all those dynamics. And of course, an individual contracting SARS-CoV-2 is not inhaling 10 to the 5 infectious doses at one shot. This is much more a mimic of what the physiological conditions would look like. And so if you look to hamsters, unlike ferrets, we do see some signs of morbidity. So hamsters at 4 weeks of age are eating and growing very rapidly. And so you can see that in a PBS treated animal, and these are cohorts of 10, you can appreciate that your average golden hamster gains quite a bit of weight over the first 2 weeks upon receipt. But if you infect these same animals with SARS-CoV-2, it's not so much that they lose weight as much as they don't gain any new weight. And so clearly they're drinking less, they're eating less, and as a result, you do have some measurement of morbidity. If you take the upper respiratory tract and you deep sequence it and look for differential expression, again, even though these are outbred animals, we use so many per cohort that we get beautiful statistics on differentially expressed genes. But this is exactly the hallmark of what we're observing in cell cultures. We observed it in ferrets, we observe it in COVID-19 cadavers. And really what you're seeing here is all of these genes, CCLs, CXCLs, these are all those chemokines I was referring to. This is the call for reinforcements. And I hope what you can appreciate is that like this whole cloud of genes that's being induced with a log 2-fold change of 6. So, this is a massive induction of chemokines, but it's happening in the absence of what you would expect. You don't see type 1 interferons here. You don't see type 3 interferons here. This is an interferon stimulated gene, but it's also induced by many other things. It's not necessarily interferon. And the interferon-stimulated genes themselves are way over here in this blue. So they are induced, but they're nowhere where they should be. They should be up here with the chemokines to help fight off this infection. And that really is part of the reason why I think COVID develops. If you take these hamsters and you follow them longitudinally, and we do some immunohistochemistry on the lungs, you can appreciate that very quickly these lungs are essentially replication factories for the virus. We see hyperplasia of all the pneumocytes. We see a diffusion of the virus across the lung as time progresses. And again, this really does speak to the fact that the job of interferon is to really contain these pockets from spreading, which is what ends up happening because the interferon system is really just not doing the job it's meant to do. The other thing I'd like to point out in this slide, which I find truly amazing, is that even as far out as 2 weeks, when we see neutralizing antibodies, the animals are fine, they start gaining weight again, even then you can still pick up very obvious pneumocytes that test positive for nucleoprotein staining. So there clearly there is sustained virus, whether it be infectious or just sticking to the residual cells. Like this might in fact explain why, for example, you can test qPCR positive for SARS-CoV-2 even after you seroconvert and have high IgG titers. And so I do think the virus is a bit sticky in that sense. In addition to looking at nucleocapsid, you can also look at just general lung infiltration by H&E. So this is what a lung should look like. This is what it does end up looking like at 4 days post-infection. So again, these animals must not feel great. This is probably why they're losing weight or not gaining weight, but overall, they do recover. This is one of the major take-home points, and this goes really well with what Carlos was saying and what he's seeing in people, is that when we look to these different tissues by differential gene expression, this is just all high-throughput sequencing, what I really want the take-home message of this to be is that we only see virus in the trachea, the upper respiratory tract here, and the lung. there's lots of virus in these two tissues. And resultingly, you have lots of pro-inflammatory signals, lots of gene movement and inflammation. But the reason I put this slide up in all this color is that in the brain, the olfactory bulb, and in the small intestine in this particular case, but we also see this in the heart, you see systemic inflammation. Even though there is no virus there, there is a lot of pro-inflammatory cytokines. There is a handful of interferon-stimulated genes. There's clearly a lot of systemic inflammation going on that goes well beyond the tropism of the virus. This is just kind of a take-home bullet point that, what makes this virus unique is not so much its ability to shut down this component of it, but it is to enhance this component of it, the induction of the call for reinforcement. And so, what can you do about this? Well, one way to do it is to do, intranasal interferon. So if the virus is in fact muting only half of the response it should be, one way to circumvent this problem would be to amplify it. And so we did this in Hamster. So this is an intranasal squirt of interferon. So this would avoid all of the systemic complications that come with interferon. Interferon induces fever, malaise, hair loss, depression. But intranasally, it's just a local enhancement of that call to arms that I was referring to. And so I hope you can appreciate that the administration of interferon to hamsters, or something that will induce endogenous interferon, does have the capacity to lower virus levels by about a log or 2. Clinical trials with intranasal interferon also look very promising. We see a reduction in total viral load in those patients that receive it, as well as a decrease in clinical symptoms and overall infection rates. It also works very well as a preventative. And I don't think I necessarily have time to go over this, so I will zip over it. But needless to say, we're also down selecting and testing FDA repurposed drugs. And we did find this one, which is very interesting, which I'm happy to talk to you about in the question period, but it's called amantadiaquin. Unfortunately, it is a derivative of hydroxychloroquine, but like interferon, in fact, as effective as interferon, it drops viral titers by about a log or 2. It makes the lungs clear up. We don't get nearly as much end staining. And overall, these hamsters, they gain weight and they look much better as compared to infected animals. Like I said, it's caused a bit of a storm because it is closely related to hydroxychloroquine. So politics kind of did feed into the science here. And so it's unclear what we're doing with the amantadiaquin results. But like I said, I'm happy to talk to you about it in the question period. And so with that, I'm just going to say thanks to all my people. I obviously don't work on the bench anymore, but my group of now about 15 or 16 people have been working tirelessly around the clock in BSL-3 conditions and building Illumina libraries and analyzing data, and they've just all been fantastic, and I thank them all for it. My general conclusions are really that I think the recipe for COVID comes down to the fact that the initial infection isn't controlled because interferon is muted, but at the same time, chemokines are just left unchecked. And that combination of those two things is essentially what results in the massive inflammation that we see systemically. Like I said, I think I covered these things. There are some hints at some successful treatments, but obviously this is still in hamsters. And with that, I'd like to thank my collaborators who have helped with all of this work, our funding agencies, and I'm happy to take questions at the end of this session. Thank you.
Great. Thank you very much, Ben. Great way to kick off the session. So again, we'll encourage everyone to continue to add your questions into Slido, and we'll come back to them as a group at the end.